agenda is an update on expanding the functionality of the CHNEP water quality trend analysis. This is going to be a presentation by Miles Medina, and we'll have the next slide, please. The purpose is to provide an update on a new trend analysis developed for the NEP Water Atlas that will estimate long-term trends and seasonal cycles in water quality dynamics. And this relates to the water quality improvement priority action number 1, assess and report water quality status and trends to identify the water quality. So, shall we advance? Miles.
All right, hi everybody, thank you. This presentation shouldn't be too long. Hopefully I can catch us up, and I'll just be giving an update on some of the work we've been collaborating on with CHNEP for trend analysis for water quality throughout the CHNEP area. Yeah, okay, next slide. So first of all, this is a huge data set, and as I'm sure a lot of you are aware, it represents a lot of monitoring effort from the last couple of decades, and effort from different agencies, SwiftMud, CHNEP and its partners, and so on. And so with that amount of data from different sources, data cleaning is very important. So the first step in this project was just to go through and apply some standard data cleaning procedures and make sure that they were applied consistently throughout the data set. And then we also applied a couple additional things. So the standard things are standardizing names, analytes, measurement units, removing data with fatal qualifier codes, removing duplicate records, field blanks, equipment blanks, and so on, and standardizing treatment of non-detects. For the most part, we were just verifying that had been done consistently. There were a couple of things that we caught that we had the opportunity to correct. And then there were a couple of additional things. So, for example, we specified some thresholds, and the Water Atlas team at the University of South Florida had already been doing this to some extent, but the idea is to, let's get rid of records that are, physically unrealistic. If you see a temperature of, 1,000 degrees, obviously that can't be right. So it's just tossing out stuff that's really obviously wrong. Then we also integrated some data that were available in WIN and STORET that weren't yet in the CHNEP's Water Atlas dataset. In particular, these were data from the strata and like Charlotte Harbor proper, like the East Wall, West Wall, Cape Hayes, areas like that. Some of these data came from SwiftMud. I believe some of them came from Sarasota County as well. And we were able to grab those from WIN and STORET to include them in our trend analysis. And then since I mentioned the strata, this actually became required a bit of effort for the last few weeks. Nicole and I had to sort of hunt down information on the strata that I'll discuss in a minute, so that we could make sure that we were treating the data correctly in terms of where it was collected, and that it was labeled consistently in terms of its spatial area. So anyway, the picture on the right is just a screen grab of the very top of the R Markdown document, where we basically document all the cleaning procedures for the CHNEP data set. There's also separate Markdown documents for the WIN data and the STORET data that we added in. And so if anyone's interested, I mean, it's a pretty long and kind of detailed document, but if you're into that kind of thing, I'd be happy to share it. Okay, so on the next slide, I'm going to get into a little bit of detail about the stratified sampling design, which is really cool. And honestly, before working on this project, I wasn't aware of the way some of these data from the estuary, different parts of the estuary, were being collected. So I think it's a really cool, thoughtful design, and we wanted to make sure that the trend analysis, the data analysis, was consistent with the intention behind the sampling design. So the way it works is that the Charlotte Harbor area is divided into these 13 strata. And the idea is that these are, more or less homogeneous in terms of water quality. And every time sampling occurs, which is typically once a month, there are 5 random locations within each strata that are visited for sampling. And so each of these 13 strata are divided into grid cells that are square mile each. And so 5 of these are randomly chosen from each strata, and that's where you go and collect your sample. And the intention is that over time you get this kind of varied and representative sampling in terms of spatially varied, so that it's representative of the entire stratum. Yeah, next slide. So this is the same picture, and you can't really read the numbers, but you see the little grid cells within each stratum. And the challenge here is that different agencies are responsible for collecting samples within each stratum. So you have Lee County in some areas, you got Sarasota County, Swift-Mud, and so on. And in some cases, the labeling of where the sample was collected isn't consistent. So like, I think, for example, Lee County uses the grid numbers, which is typically like a 3-digit number, like 193 or something. And that gets assigned as like the station ID when the data get put into Win and Storette, for example. But then if you're Sarasota County, you have completely different labels for the locations, and they have nothing to do with the strata or the grid numbers. And so it's not obvious. And we had to kind of build this spreadsheet of associations between what people are calling their sample locations and how they relate to this stratum design. And so this is really important work, because without knowing, like I did a few months ago, that these strata exist, that this stratified design exists, and then even if you know that, if you don't know what the associations are between how the data appear and are labeled in a database like Win Storette and what they actually mean in terms of this stratified design, you just can't analyze the data according to that design. So I think this is a really important contribution to taking this sampling design a step further into the data analysis. And so, it improves our trend analysis work, but by making this data set public, I think it's also going to be a good contribution to analysis that others can do as well. Okay, so next slide. All right, so moving on to the trend analysis. So once we got the data cleaned up, we found that we had 555 different locations. So there's 542 stations, like traditional point stations, a particular point along a stream or a river where sampling occurs. And we have those 13 strata polygons. Each of these locations has up to 17 analytes. So we've got nitrogen and phosphorus, different forms of those, chlorophyll, oxygen, color, and so on. And for this project, we focus on a 10-year period between 2011 and 2020. In the future, we're planning to extend this back to the year 2000 and to bring in the last couple of years of data as well. But for now, we're just looking at this 10-year period, the tens. And the trend analysis method is really something that's pretty cool. If you're not familiar, Marcus Beck and his co-authors, Marcus Beck from Tampa Bay Estuary Program, published this paper back in January of this year. And it introduces a really sophisticated and I think much more realistic and nuanced way of looking at water quality trends than the sort of standard ways like Kendall. And so to break it down, there's kind of 2 basic components. First, we use generalized additive models, or GAMs, which allow us to fit it's a very flexible form of regression. So we can fit trends, whether they're linear or nonlinear, over the long term, and we can also capture seasonal cycles. And the seasonal cycles are allowed to vary. So there's an interaction term between the long term and the seasonal. And so seasonal cycles don't have to, be modeled the same way every single year. So there's a lot of flexibility in there, but it's also penalized so that you're not overfitting. And then based on the GAM fit, and I'll have visualizations for this if this isn't clear. But based on the GAN fit, then we can use mixed effects models to estimate the trend over a specified period. We do 5 years and 10 years, but you can pick any time frame. And that gives you the traditional kind of up or down determination, whether things are significantly trending up or down. And a nice feature with this mixed effects models approach is that it accounts for uncertainty that arises from variability and sampling effort over time. So if you had a particular year where there was relatively less sampling, that gets accounted for, and you have basically more uncertainty, a wider confidence interval, and so that ultimately has less of an effect on the up or down trend determination. So if you're not aware of this paper, I recommend looking it up, or I can send you a copy. Really cool stuff. Okay, next slide. All right, so to explain a little bit more how the trend analysis works, I've got 3 slides with some examples. These are results. So as you might expect, temperature is often kind of the simplest dynamics. So here you see the 10 years from 2011 to 2020, and in the top panel we've got the data in the gray points, and the GAN fit is the blue curve, and you can see the seasonality there. But you also see things kind of trending up just from the GAN itself. The highs are getting higher, the lows are also getting higher. The bottom 2 panels are the results of the mixed effects model. So the blue bars show the mean estimate for temperature for each year. And then we have a confidence interval around that. And here the trend results are pretty straightforward. I mean, it almost falls along a straight line. So you see the red trend line for the 10-year and the 5-year periods from 2011 through 2020, and then 16 through 20. That also has some uncertainty around it. That's the pink band. And so, this is a very straightforward example. You see temperature consistently trending upward. Next slide. Now here is just wanted to give an example of, slightly more complicated case. We've got a GAN curve in the top panel that is a bit more wiggly. There is some seasonality, that's really apparent in some of the years, but there's also some additional wiggles on there, and the seasonality you can see varies. There seems to be less of an oscillation in the latter half of the period here. But nonetheless, we still see a pretty consistent downward trend in the mean turbidity in this case over the 10-year period and the 5-year period. And yeah, okay, so let's go to the next slide. All right, and then here, this is a much more complicated example. Not only is the GAN very wiggly, this is dissolved oxygen concentrations at the east wall of Charlotte Harbor. But we see something interesting, and this, this is something that if you've done water quality trend analysis before, you've probably thought about, where your typical Mann-Kendall analysis is looking for a consistent kind of up or down trend, right? And what we see in the second and third panels is the means for each year are sort of all over the place. The first 3 years, it seems things are sort of trending up. Then it kind of resets in 2014, it goes up, and then it comes down again. And so to capture that complexity, here we're only looking at 2 time periods, the 10-year time period and then the 5-year time period. But as they discuss in the paper, you can really pick any time period you want. I mean, you could go down to 3 or 4 years and then use like a sliding window and see how those trends evolve over time. And they propose some different plots for visualizing that, to see if, I guess, how robust the trends are to the selection of the length of that window. So if you're looking over 10 years here, it's kind of glossing over a lot of variability. And so some of the ups and downs end up sort of canceling each other out, and you get this kind of flat curve. By the way, the gray indicates that it's not a significant slope. But then if we look at the 5-year, at the bottom, we see that there's a downward trend. So it's helpful to look over different time periods. And here we're just looking at 2 different time periods, but even that is pretty informative. I also want to note that we're looking at mean annual values here, but you can really use any metric. I mean, you could use the median, you can use min or max, you can even restrict instead of doing an annual mean, you can do like a seasonal mean. So if you were looking at chlorophyll and you wanted to look at, like the kind of summertime average or high, you could base the trend analysis on that kind of metric as well. So there's a lot of flexibility here, based on kind of what questions you want to ask. It's a really cool framework. All right, so next slide. So what I showed up to now are results for particular analytes at particular locations. But of course, like I said, there's over 500 different locations we looked at. And these results, these maps that I visualized, I just generated these earlier this week. So if you'll bear with me, I think the aesthetics still need a lot of work, but this is the base map I used, and you'll see in a moment why it shows black and white. But even just starting to look at the results and try to digest what's going on, there's some interesting insights, I think, so far. And if we have time, it'll be interesting to see what people make of these based on their local knowledge and experience. Okay, so next slide, we're going to look at total nitrogen. What we're looking at here is we see both points and polygons. So the polygons are the 13 strata. So we've got tidal Peace River, tidal Mayaka, East and West Wall, Charlotte Harbor, Lower Charlotte Harbor, and so on. These are colored red, yellow, or green to indicate trends up, sideways, meaning no trend, or trends down. On some of these slides, you might see some gray polygons, and that's going to indicate that there weren't sufficient data to do the trend analysis for the given period. And so that's the polygons. We also see points, and these are for the point stations, up or down triangles in red and green, respectively, for the trends. Little yellow dot means no trend, and gray dot means not enough data. So in this side-by-side comparison, we've got on the left the 10-year period, 2011 through 20, and on the right, the 5-year period, 2016 through 2020. And I think what really stands out, if we just look at the polygons, we see a lot of the red strata have moved into the yellow, which is a good sign, because it means over the 10-year period, things have overall increased. The concentrations have trended up. But during the 5-year period, it's kind of flattened off. And if you look at the individual results for each stratum, you see that means that the concentrations have sort of leveled off on average, which is hopeful. Now, on the other hand, it's not just about trends, which are sort of relative. You also have to look at the actual concentrations relative to some threshold. And so for total nitrogen, from what I've looked at so far, it seems that even though it's good news that the nitrogen has sort of leveled off the last few years, the concentrations are still pretty high relative to the estuary specific criteria. So in a lot of cases, they're still in excess of those criteria. But the fact that things have leveled off is good news. Yeah. Okay. So let's move on to the next slide. That's chlorophyll A. Here, it's a similar story. We see some of the red, like Pine Island Sound, has moved into the green, meaning that over the 10-year period, things have trended up, but they've kind of changed direction more recently, and they've started trending down over the last 5 years, 2016 through 2020. We also see some red moving into yellow, which indicates a sort of leveling off. And here, when you look at the actual values, concentration values, seems like in many cases, the averages are at or below the estuary specific criteria. So really good news there. There are a couple of gray areas in Charlotte Harbor proper, so the tidal river areas, the east and west walls, and Cape Hayes. And the reason is that the data that we have, that we obtained from Wynn and Storette, it goes back to at least 2011, almost certainly before that, but we've got data, from the beginning of our analysis period, and then it ends in the middle of 2019. So we didn't have the 10 years to do, or the 5 years to do the trend analyses for these areas. Now, I'm talking with Nicole. I'm pretty sure that these data exist. They're probably just, SwiftMud has them, and they haven't been loaded into Win and Store yet. So that'll be something to work on in the future. So right now we don't have trend analysis results for those areas, but where we do, the Lower Harbor and around the Caloosahatchee and so on, the results are actually looking pretty good. And then finally, on the next slide, we can look at dissolved oxygen. So here we see some trends going in the other direction. We see some yellow moving into red, green and Matlacha Pass moving into yellow. And so things are going from relatively stable over the 10-year period to the dissolved oxygen concentrations going down. Here the colors are flipped because obviously lower dissolved oxygen is less desirable. Maybe a little bit of bad news there in terms of the trends, but in absolute terms, the concentrations by and large are still on average, annual average, still pretty high, around 5 or 6 milligrams per liter. So I think these are just some empirical results. Some gives an impression of what the conditions have been over the last 10 years and 5 years. What explains these trends? I think that's a good question for further research. Is it mitigation of efforts, restoration efforts? Is it something biological going on, seagrass, macroalgae, things like this? I mean, these are all interesting questions that I think we're going to want to look into a bit more deeply. And if anyone has any anything to discuss or any ideas there, I looking forward to hearing that. Let's move on to the next slide. Okay, so next steps for this project are, as I said, to refine the map aesthetics. I don't think they're very pretty. It's kind of cluttered because there's just so much information on there. So we're going to have to workshop that a bit. But I did want to share, where we're at today. And then finally, we're also going to design a web-based dashboard to showcase these results on the Water Atlas website. And on the right is just a kind of a crude mock-up for what we imagine this can look like. Not too different from what's currently up there. We'll have a map with trend directions and so on, like I showed in the previous slides. And then on the right, we can show, the time series, the GAM curve, and the trend, trend direction for 10-year and 5-year periods. All right. So yeah, next slide, final slide, just contact information if you have any questions or want me to share any of the materials that I showed you today. And if there's time, I'd love to hear, anyone's thoughts on the work and what it might mean. Thank you.
Great. Thank you so much, Miles.
Can I just say before we get into discussion of this, I think that this work is really a huge testament to all of the agencies and CHNEP way 20 years ago who got together and decided to do some of this sampling and identify data gaps. And I think it's really cool to see some of this come to fruition in this way. And thank you, Miles, for presenting it in this sort of really cool, intuitive way and doing this new style of analysis. But I wanted to just thank some of those people are since retired, but all of those people that were involved in those decisions and, the sampling, design, and collecting the data over 20 years. I think that's really cool. So that I wanted to open it to the floor.
Any comments from the board here? Yes, Mindy.
Miles, can you hear me?
Yes. Hey, Mindy.
Hi. Great work. I think it's really good. So this is 2 questions. I guess this is just analyzing the CHNEP water quality data, and that will be on its own page on the Water Atlas.
This will be the trend analysis page on the CHNEP Water Atlas. And it is, so Miles kind of highlighted the work that we've done to pull out the CCHMN strata data, but actually he did the analysis on like each of the stations collected by the Chevy Walkman program and any of the watershed sampling stations that, like Lee County has watershed sampling stations, eventually Charlotte County will, Sarasota County, those are all on there as well.
Okay, cool.
Is that correct?
And then my other question was, like you said, when you were presenting, so you're gonna have the trend analysis for each of the parameters, but if it's still above the numeric nutrient criteria, I think that would be interesting to plot. So maybe if you plot the annual averages of like nitrogen, for example, yeah, by year, and then have that numeric nutrient criteria as a line. So even if it's going down, you can still see like, yeah, it's still above that target. That would be helpful.
Yeah, totally. I agree. I think USF's page might show some of that, but I think that's definitely an important feature to have. And like you said, it's easy to implement because it's just it's a line. And for the strata, there's those estuary specific criteria. For other locations, like especially throughout the watershed, there probably aren't criteria, but you can adopt something like class 1 or class 2 or class 3 kind of standards, and because it's not a regulatory an explicit regulatory exercise, I think as long as you're transparent about what you pick as a threshold, and I think that's fine. And just to add more broadly, like our work at CCS, I mean, we're interested in identifying, I'm sure like all of you are, identifying priority areas. And so I think combining the trend information with a more absolute kind of information above or below some threshold, for example, then you end up with this kind of matrix of like tiered priorities, where the highest priority tier would be it's above the threshold and it's trending up, right? And so all the combinations of the absolute and relative to the threshold, and then the relative categories that you get from the trend analysis.
Yeah, this is Jennifer. I'm going to jump in really quickly because this is actually, what Dr. Medina and the team at UF has done is a puzzle piece and a larger effort that we have undertaken. We have 3 staff working pretty heavily on this right now in the CHNEP staff to overhaul the water atlas. So we have really looked at, over time, we've developed a lot of tools that are operating in parallel on the water atlas. What we're trying to do is integrate those all to be water body pages, basin pages, watershed pages, and to aggregate that. And we would have 4 interactive mappers: water quality, hydrology, fish, wildlife, and habitat, and climate change data. And then we also will have these tools that he just mentioned, like the dashboard feature, which is going to take every water body and look at what criteria, and it's going to start with, is there a TMDL? Is there site-specific alternative criteria? If not, then you go to, generic criteria or whatever for that classification of water. So we've created flow charts, and we are working with UF and USF to basically rebuild the whole site to be more integrated and holistic. But in doing that, we had to go back and look at pieces like this trends analysis, where we were seeing things that needed to be improved before we can integrate it. So that is what we have done here, is really update using the latest, most sophisticated analytical approach, getting the data as clean as possible, so that we can get the best tool to then put into this new updated water atlas, which we should be starting to roll out. I mean, we're rolling out pieces of it, but the new water body pages will be completed the end of next fiscal year. So by the end, this time next year, we should see a really different water atlas. But I know there was one comment that came into line. Judy asked or suggested that it would be helpful to show these trends compared to criteria. These estuaries are all outstanding flow to waters, which is another good point. It is possible that a 5-year good trend might be misleading if the values are already higher than the OFW levels. So we will make a note on the water body pages of which water bodies are OFWs. From a policy perspective, that doesn't necessarily mean that, unfortunately, even though in theory they're supposed to be protected against any degradation at the time of their designation, a lot of them were never sampled at the time of their designation. And so there is no baseline from a policy standpoint, that would establish whether degradation has occurred. And we've argued, or I have in my previous capacity argued to DEP about if you see degradation occurring, it doesn't matter when the baseline was, but nonetheless, a lot of times they are still being handled essentially the same as whatever classification of use they are. So the dashboard is going to be using what the DEP applies as their criteria, if that makes sense. But to Dr. Medina's point, it's not trying to supersede the impaired waters rule designations or anything like that. It's just to show what is the most real-time data show us, and there'll be dials like green, yellow, red. What is the most real-time data last sampling show us as to where those levels are for these different analytes versus what the criteria is for that parameter. So it's going to be a helpful tool, we think, in kind of keeping a more real-time pulse on the health of these systems. But I think the last thing I'd mention is that what I saw someone say about chlorophyll, like eyes got eyebrows raised and stuff like that. I think that the problem is that there's so many different things going on at once, and the macroalgae and the seagrass loss is definitely a player in some of these systems. And so what we're trying to do is bring that into the water body page so that we can start to see how all these things intersect, so that we get a more complete picture of what's going on.
This is Rick. I have a comment.
Go ahead.
The one problem with using the water quality is if you have large mats of macroalgae, then that can affect the water quality and maybe make you see better water quality than you would otherwise see if the macroalgae mats weren't there. For example, Mindy and I have been seeing about a metric ton of macroalgae per meter square, about a kilogram of macroalgae dry weight per meter square in Charlotte Harbor and Matlacha Pass. That basically is a trap for turbidity. Particles settle out into it. It improves the DO for part of the year, and it's going to decrease the DO as it decomposes for another part of the year. So you may not see an overall trend in DO, but if you look at the low DOs, then you'll see a trend to having more low DO days. And then, for example, nitrogen concentrations are dependent on the amount of uptake of dissolved nitrogen. A kilogram of macroalgae can take up a large amount of nitrogen, more than you can put in from the watershed. So it's getting nitrogen from the sediments, nitrogen and phosphorus from the sediments. So the 10-year and the 5-year graphs you showing increases in improvements in water quality could just be from having more macroalgae, more nutrient demand by the macroalgae, and having less chlorophyll in the water column because there's no nutrients in the water column for phytoplankton because the macroalgae are taking it all up. So we're seeing pretty large changes in macroalgae since 2018. That's going to improve the water quality for part of the year. Just looking at water quality trends isn't going to show what the problem is or what's happening very well.
Yeah, thank you. I agree completely. It's it's part of the story, and I think the results always have to be interpreted with caution. There's a broader context, right? So I think part of the solution there is, and I know Jennifer, your Water Atlas has information about the biology as well. So making sure that that's, I think, kind of like carefully, thoughtfully communicated so that results aren't taken in isolation and then misinterpreted. I think that's really important.
Hey, I think thank you so much, Mike and Miles, sorry. And then we are ready to go into a break. I highly recommend, encourage you guys to stay around after